So without any further ado, let's turn to our speakers. Dr. Issa will be first, and welcome Dr. Issa to the podium.
Thank you. So hello, everyone. Thank you, Andy and AdvaMedDx, for inviting me. I'm delighted to be here this afternoon. I'd like to start with a very brief story. A woman is driving here in Washington, D.C., and realizes that she's lost. She pulls over to the side and sees a man sitting at a bus stop, and she says, "Excuse me, sir, can you please help me? I was supposed to meet a friend about an hour ago, but I'm completely lost. Can you help me out? Can you tell me where I am?" And he says, "Sure, I'd be happy to help. You are at 38 degrees longitude and 77 degrees latitude, and you're driving a black BMW." She looks at him quite perplexed and says, "Are you a researcher?" He's surprised and says, "Yes, I am. How did you know?" And she says, "Well, what you've told me is technically accurate, but it's absolutely of no use to me. I'm still lost, and if anything, I'm even more late than I was before." And he looks back at her and he says, "Are you a policymaker?" She says, "Yes, how did you know?" And he says, "Well, you have no idea where you are or where you're going. You made a promise that you can't keep, and you're still exactly where you were before, but now you're blaming me for it." But in all seriousness, it's wonderful to be here because over the past several years, researchers and policymakers have actually found ways to work very well together and have some very fruitful relationships and exchange. So I'm really delighted to be here today. So what I hope to do in the short time that I have with you this afternoon is to explore these two questions. What do we know about the adoption of molecular diagnostics in clinical practice, and what needs to be done for molecular diagnostics to be more routinely adopted into clinical practice? And to show you why molecular diagnostics, and particularly molecular diagnostics as they are being used in cancer, because that's the theme for us this afternoon, are making all the difference in the world for patients right now. So molecular diagnostics are, as Andy said, are tests that identify molecular and genetic markers for an individual patient. They can determine potential benefit from a specific therapy, or they can determine risk of developing a specific disease or health condition, and they're also used for various other purposes as well. So let me introduce you to Betty. Betty was diagnosed with breast cancer some 20 years ago in 1992. Now, what happened when she was diagnosed is that she first received a mastectomy. Then she underwent chemotherapy and then radiotherapy. And all of this, what we now might think of as a shotgun approach to treatment, was at the time standard of care for breast cancer. Now, this is Evelyn. Evelyn was diagnosed with breast cancer about a year ago. Unlike Betty, who was diagnosed 20 years ago, Evelyn had some options. She might have had her tumor profiled to better understand what drugs might work best for the particular type of tumor she had. Her tumor was profiled using one of the newer gene expression profiling tests to understand how aggressive her cancer is and whether it is likely to recur or not. So Evelyn will have had a chance to be treated the right way the first time, right from the get-go. The options are there for her. And this is increasingly becoming current practice. And it is also the future of personalized medicine. And these different components are coming together in a way now that is leading us to ask some fundamental questions about how cancer is characterized and treated. What do we need to know? When do we need to know it? And what difference does it make? What do we need to know specifically when someone is diagnosed with a particular disease or cancer? When do we need to know it? What difference does it make? When do we need to know that you have a disease like breast cancer or colon cancer? What does that mean for you as an individual? And what does it mean for your treatment? And so we're beginning to move towards this new concept, a new era, if you will, of personalized certainty, knowing with a greater degree of accuracy that was not available to us before, a degree of precision that's more in line with what patients and their families and their healthcare providers want. That's appropriate for these types of life and death decisions, providing us with more right treatments for specific subpopulations and individuals than ever before. And this is driving new value propositions that have been unimaginable until now. And this creation of value is what is driving the field of personalized medicine forward. Well, all of us are different. We all know this. And you all know that what's behind our differences in eye or hair color or height or what have you is largely DNA. The 3 billion pieces of data in every cell of your body that is the blueprint for all sorts of things that make you who you are. Whether you are tall or short, whether you have blonde or brown hair, and also how you might get certain diseases or not, and how well you might respond to particular treatments or not. So given that we all understand this and agree that our DNA is largely responsible for our differences, why is it that up to fairly recent times we have been content with a one-size-fits-all approach to therapeutics and disease treatment? Because the fundamental reality is that most diseases are considerably more complex than the size of your feet and whether your shoes fit or not. So you would think that before we can truly understand diseases, particularly something like the many cancers that befall us, and we are all, in one way or another, affected by cancer, because if we don't get it ourselves sooner or later, we have a friend or a loved one who does. We ought to have a good idea of what's going on and what goes wrong at the level of complexity that's appropriate to the disease, and that is often at the molecular level. So when you hear the term diagnosis, if you're like most people, you tend to think of it in somewhat of a reductionist way, quite simply as sort of a binary concept. I am healthy, I'm not healthy. I have cancer, I don't have cancer. I feel great, I don't feel so good. But over the past several years, we've had tremendous advances in the field of molecular biology and in genetics and genomics. We now have technologies such as DNA and whole genome sequencing, polymerase chain reaction, PCR, protein analysis, and proteomics. And all of these mean that we now understand disease at a deeper, more fundamental level than has ever been possible before. So we're beginning to understand at a much deeper molecular level what really makes us sick. And now the really interesting thing is that these research tools, which you see here, which for many people, like many of you in this room, are sort of abstract because they don't necessarily see them. You typically think of them as these cool toys that scientists play with in their research labs, are now increasingly being transferred and translated from the research labs to the clinic. So instead of simply being able to understand what makes a disease, we are now able to apply these types of powerful, sophisticated technologies and tools and understand what makes your disease a disease. And this ability to analyze and characterize individual patient genomes is making all the difference to how we treat your disease. What choices are being made among an increasing array of treatments to improve outcomes? So, going back to my earlier point, you can see that the term diagnosis, in the way that most of us think of it, really is quite a reductionist term, because it doesn't capture the complexity of disease, and it doesn't capture the fundamental interdependence between all the genes and proteins and other elements that make us sick. So molecular diagnostics are giving us a new ability to characterize disease, and this is a growing area of research, and it's really changing how we think about where the field of personalized medicine is going to be effective. So let's go back to Betty again. When she was diagnosed about 20 years ago, she had to go through a mastectomy with all the trauma and the difficulties associated with that. Then she had to do a round of, perhaps maybe several rounds of chemotherapy. And then she underwent the radiotherapy. And the interesting thing is that we now know that only about maybe 20–25% of women are going to respond to chemotherapy depending upon the type of cancer they have. But we had no idea, especially back then, who the responders were going to be. So everybody got chemotherapy. as a matter of course, as a standard of care. And as patients and as a society, we simply had to live with the consequences of this shotgun approach, with the side effects of chemotherapy and the costs. But let's take a closer and deeper look at breast cancer. This is what I, as a scientist, and my colleagues think of when we think about breast cancer. This shows what are called signal transduction pathways. So this shows a whole group of proteins and genes, and these are the molecules that convey the messages inside the cells and also between the cells, and there's a lot of crosstalk going on between them. Someone once described this kind of diagram as an electrical wiring diagram gone horribly wrong. But when you see this, you can begin to think of cancer, and this applies really to any cancer, not just breast cancer, but you can begin to think of it as a very complex, incredibly interdependent wiring diagram, perhaps the most complex wiring diagram you might have ever seen in your life. So this is the sort of diagram that we use to describe signal transduction pathways, and this is actually only one of the pathways that may be relevant to breast cancer. So we are increasingly understanding how changes and defects in these molecules drive particular diseases. And equally important, what we are also learning is that breast cancer and many other cancers, it's not just one disease. There are a number of different subtypes of breast cancer. And this is true, by the way, as I said, for all kinds of cancers. There are often many subtypes of those. And so, in fact, scientists and physicians now tend to describe cancers by their molecular structure rather than by their location. So it makes more sense nowadays to refer to KRAS as a cancer, for example, than to simply talk about breast cancer or prostate cancer or lung cancer. Because that makes more sense to describe what's gone wrong, and it starts to lead us in the direction of where we're going to apply the therapy or the treatment. So if we understand at a molecular level where the fault may lie, then that will direct us to where to apply the therapy, the treatment. And so understanding these mechanisms of action and being able to make deeper diagnoses at a molecular level is an absolutely fundamentally critically important area of research. And when it comes to cancer care, the numbers are staggering. Tens of billions of dollars are spent each year on treating cancer, in this country as well as around the world. Now, if you're still thinking in terms of a one-size-fits-all approach, then first of all, you're not helping the patient, and secondly, you're not doing the healthcare system any good. And we know from studies that have been done that billions of dollars are wasted in the US and in other countries every year on treatments that make no difference to how cancers are being treated. So personalized medicine and the use of molecular diagnostics can not only benefit patients, but also are poised to realize some significant efficiencies in the healthcare system. So let's go back to Evelyn. One of the first things that happened when Evelyn was diagnosed with breast cancer last year is that she had her tumor profiled using molecular diagnostics. That profile turned out to be ER positive, PR HER2 positive, and that would have been immediately and completely relevant to the first line of treatment that she received. And then she might have had her tumor further characterized using gene expression profiling tests, such as OncotypeDx or MammaPrint, which would indicate the likelihood of cancer recurrence. And these tests would have given her a recurrence score. And so within a matter of days, she and her healthcare provider, her physician, would have a better idea of whether she was going to need adjuvant chemotherapy following surgical resection of her tumor or not. So this type of information is valuable for Evelyn and other patients like her in that it can make all the difference in the course and progression of her disease. It can make a difference as to whether Evelyn undergoes a taxing course of chemotherapy treatment, whether she and her family incur the expense of chemotherapy, and whether she loses her hair or not, and is subject to sometimes unnecessary toxic side effects. So as we look to the future for Evelyn and other patients, they are likely to have options, choices in treatments that will make a difference to the right, in terms of getting the right treatment to the right patient at the right time, personalized treatment for their cancer. And this is the fundamental value proposition of molecular diagnostics, providing these options. And this is also making a difference for the pharmaceutical industry and for drug development. If you think about how we're beginning, how clinical trials are being conducted, we're beginning now to increasingly see the stratification by subpopulations of patients in clinical trials. And this is leading to the repurposing of drugs that might not have worked as well before, but now we can repurpose them for another use, relabeling of drugs by the FDA. And over the past year or so, we've been seeing some significant launches of drugs for a number of cancers like melanoma and lung cancer, and you'll hear a little bit more about that shortly, and also prostate cancer. And all of these really are beginning to take advantage of the power of personalized medicine. So we are beginning to use these tools, these biomarkers, to select which patients to include in clinical trials and in drug development, and we are beginning to get drugs to the market a little bit more efficiently. Well, this is all very well as long as you're using molecular tests to rule in medicines or drugs. But what happens when you think about ruling drugs out? Now we have to start thinking about some other factors as part of this equation. And this becomes incredibly important because if you're going to use these technologies, these molecular diagnostics, these tests to determine which therapy should be given, and importantly, what therapies should not be given, ruling drugs out, then you better be sure that you're making the right call. And particularly in the case of cancer, where we're talking about life or death decisions, you want to make sure you're making the right call. And this is where the complexity of decision-making comes into play. So you will want to make sure that the test is validated. And we have all sorts of sophisticated tools from epidemiology and from clinical trials to help with that validation so that you have the confidence to make that call and be able to say, this treatment will not do this patient any good. And you're also going to want to make sure that the test is regulated, and that's what the FDA is for. You'll also want to be sure that the provision of this particular test is commensurate with the degree of accuracy and precision that is required and expected in healthcare in our society today. And you'll want to make sure that these tests and the consequences of their application are going to be interpreted correctly for patients in order to be able for them to make good and sound decisions. So as we are increasingly crossing this translational divide from research to real-world practice and implementation in clinical settings, what we're finding is that all of this is leading to a whole set of new questions. Questions that are making us, as a society, feel as if we're sailing in as yet to be charted waters. Questions related to analytical validity, and clinical utility, whether the right clinical needs are being addressed and being met, whether the applications are acceptable to patients and providers. And these larger health system level issues are exactly the types of questions that we at the program in personalized medicine and targeted therapeutics work on. Our research agenda for some years now has been largely driven by the question, what is needed for personalized medicine to be translated and well integrated into healthcare delivery and health systems? And so one line of investigation, just as an example that we are very active in, has to do with decision making and how patients and providers are using these new molecular diagnostics to make better decisions about treatments. And we have developed instruments to quantify, to measure how patients make decisions about using and adopting molecular genomic diagnostics, and how they make trade-offs between the options that are presented to them. Because as I hope that I have convinced you, as we increasingly use these sophisticated tests, we also increase the complexity of decision-making in clinical settings. So why is this important to investigate? Because for the longest time, since the completion of the Human Genome Project, we've been accumulating lots and lots of data, about 3 billion data points that make up our human genome. But now we've reached the tipping point where we're actually accumulating more comprehension to understand what that data means for us. And with that increased understanding comes the complexity of decision-making. Because if you think about it, if I've convinced you that by now that cancers are complex and that they need complex interpretation, what would that change for you? What would you do with the information that you get about your disease? How might that change your behavior? Because if there's anything that's more complex than signal transduction pathways, it's actually human behavior. This is an artist's rendition of what Michelangelo's famous statue of David might look like with a higher BMI. So just to give you a very quick example of some of the work we do, this is from a recent paper in which we modeled some of the decisions taken in breast cancer treatment and compared two commercially available gene expression profiling assays, OncotypeDx and MammaPrint, including quality of life and cost-effectiveness issues, and we found an unexpected result. We found that OncotypeDx, which is the most commonly used gene expression profiling test for breast cancer in the US, is actually the test that's less cost-effective, and also it exhibited slightly lower quality-adjusted life years, QALYs. But what's interesting about this is not so much just the results, that is in and of itself interesting, but the fact that policy decisions by payers and others had been made in the absence of this type of evidence. So this was really the first study of this type. And so there's a real need for more translational research studies of this type to be conducted alongside the molecular biology research as well, as we, as a society, continue to implement novel genomic molecular diagnostics into practice. So I've told you about what molecular diagnostics can do and how they are being used for risk prediction, screening, diagnosis, and therapeutics. And I've talked about the value proposition. But the speed with which and how well we manage to adopt new molecular diagnostics into practice hinges on several factors. Certainly decreasing costs, which we're beginning to see in terms of sequencing and technology. The sequencing instruments are now clinically affordable. Increasing the speed of sequencing, which now we are able to do it within a 10 to 36-hour window. Bioinformatics. This is still very much a roadblock because we still need to be able to figure out better mechanisms, both in terms of bioinformatics, but also electronic medical records and electronic health records for how to do this better. Clinical usefulness, again, this is going to, as more of these tests are proven to be clinically useful, more and more of them are going to be used. We're still dealing with the roadblocks of payment uncertainty because payers do not fully understand genomic analysis, as well as regulatory uncertainty. So these are some of the issues in addition to the continued research, molecular research, that we also, I think, need to deal with in order for this to become realizable, a reality. Today, we really are at a tipping point. We are able to characterize disease at the molecular level more accurately, leading to increased efficiencies, better therapeutic responses, and reduced adverse events, and diagnostic and risk prediction processes. And all of that together is going to help us, has the potential to transform healthcare because it all makes a world of difference to patient care now and in the future. Thank you for your attention.